chapter 24 from your textbook, and that is on the biosynthesis of amino acid. Biosynthesis of amino acid, because amino acid turnover and protein turnover, which we studied last class, and now I would like to go over to the biosynthesis. The biosynthesis here, it involves the amino acids, meaning there is an amine group is there, amine group, that is NH2 group, amine group. And this is the basic structure of or any biological macromolecule. You need to have the amino acids in order to synthesize the other biomolecules. So the amine means, if you see the structure, you have a nitrogen and hydrogen. So where this nitrogen is coming from, it's coming from the atmosphere. Okay, it comes from the atmosphere, the nitrogen. And they are fixed, the atmospheric nitrogen is fixed by bacteria. Okay, so you have to, the nitrogen from atmosphere is fixed first, fixed by bacteria. Why? As I mentioned earlier, in a Calvin cycle, the carbon dioxide, CO2 fixed by plants by photosynthesis, right? Now the nitrogen from the atmosphere, this is also from atmosphere, this is also from atmosphere, but the plants and bacteria can photosynthesis, fix carbon dioxide as a food. Now, if you want amino acid, the amino acid also coming from the atmosphere, that is fixed by bacteria exclusively. Because there is not a plant is not fixing, or the humans cannot fix in the nitrogen, but the bacteria can fix it. So now we are depending upon the plant for carbon dioxide and carbon skeleton, and we depend upon bacteria to fix the nitrogen. Now the next level is, we know about the carbon dioxide that leads to produce sugar molecules with the carbon skeleton. Now the nitrogen is fixed by bacteria, that will be used for amino acids, and amino acids to protein. So we'll see how the bacteria will convert the atmospheric nitrogen. Okay. So how much nitrogen is there in the atmosphere? 78% approximately, if nitrogen from atmosphere. And you have 20% of oxygen. Okay. So 78% of nitrogen, the atmosphere air is composed of the, you have this nitrogen, and 20% is an oxygen. And the rest of them, the other gases and carbon dioxide and other gases also there. So now the bacteria, okay, where the bacteria is present is bacteria, they are fixing in atmospheric nitrogen to the soil, and some other bacteria which is present in the plant root, especially the legume plant roots. Okay, plant root nodule, plant root nodules, and they fix the atmospheric nitrogen by the enzyme system. Okay, how they do with the nitrogen, and they convert to ammonia, and then this ammonia is NH3, and then it is going to convert to glutamic acid or glutamate, first amino acid, glutamate, and then from the glutamate other amino acids are being produced from this end. Okay. So as I mentioned earlier, the assembly of the biological molecules, assembly of biological molecules or biomolecules. Biomolecules of what? Proteins and nucleic acids. These are the biomolecules. Okay, and they require a starting material. A starting material. So the starting material is from amino acids. Amino acids. Okay. So how these amino acids will be produced now? We have already studied some of the energy transduction pathway, like a TCA cycle. The other pathway, that is, we call it as a glycolytic and TCA cycle, and in other words, they produce more of energy. So we call it as the energy transduction pathway. Energy transduction pathway, and the pathway, they are enriched large amount of intermediate compound. If you remember, there are different steps, and to end with the carbon dioxide, and there are a lot of intermediate compounds. Intermediate compounds. And these intermediate compounds are responsible for production of amino acids. Okay. So we are going to see one by one. Okay. That's the starting material. This is the starting material for amino acids. Where energy transduction pathway. Okay. There are a lot of intermediates in this pathway, which is TCA cycle, glycolytic pathway, pentose phosphate, shunt, and other pathways. You know, you have all the intermediate. From that intermediate, we can get the amino acids, which is the building blocks of certain proteins. Okay. The degradation of catabolism of the glucose or other material. Okay. Even degradation or catabolism, degradation of if you have carbon skeleton, otherwise is glucose and other carbohydrates, that includes degradation of glucose, that also includes some of them are the fatty acids. That's also been degradation pathway, and all of them, they provide intermediates for amino acids. I put short amino acid, and nucleic acid I put Na, biosynthesis. Okay. So also the nitrogen, that is the source for nitrogen. Nitrogen is important source for what? Nucleotides or nucleic acids, nucleotides, DNA, RNAs, and neurotransmitters. Now, which is a neuron, one neuron to the other neuron, they are talking the language. There's neurotransmitters, they are the signal molecule from neuron. And prosthetic groups, there's an active group in the enzyme, prosthetic group, like a porphyrin, P-O-R-P-H-Y-R-I-N, porphyrins. Okay. And the amino acid biosynthesis is intimately connected to nutrition. Now the next step, amino acid biosynthesis is intimately connected with, they are intimately connected with nutrition. Why? Because the amino acid, there are 20 different amino acids which is form different kind of polypeptide to synthesize protein, but in this case some of the amino acids are certain amino acids which we cannot synthesize in our system. As I mentioned earlier, the degradation of the glucose and fatty acid, they produce amino acid or biosynthesis. There are certain number of amino acids, you know, among 20, there are certain number of amino acids we cannot get or synthesize in our body. So we have to depend upon the nutrition or diet, whatever the dietary origin, or that's very important for our health. So they are called the essential amino acids. We are going to see that. Okay, why the nutrition or diet is very important for the amino acids or the biosynthesis here. Okay. Then some amino acids, okay, diet can provide, as I mentioned, but some of them are synthesized in our body. Okay, we will see one by one. That's biosynthesis enzyme. If you have biosynthesis, the enzymes is involved. That's what enzyme. Okay. And these enzymes which I mentioned earlier, that start with microbes. Microbes is the one which will fix the atmospheric nitrogen to ammonia. Okay, so we will see that one, microbes, the enzyme system in the microbes. And then we can also see plants. Plants also have the enzyme capability to fix the microbes, fix the nitrogen to ammonia, and this ammonia as a fertilizer to the plant, and the plant will produce the ammonia to amino acids. Ammonia to amino acids. The plants will do that. Okay. So if you have some antibiotic, okay, if you want to kill only the microbes, you can choose the enzyme inhibitor, and thereby it can inhibit only the microbe, not for the human or not for the plant. So there's some antibiotics can be produced, and certain type of plants which you don't want to get some of the weed and weedicide like. So those things are exclusive of the plant origin. So you can use that particular inhibitor, and thereby you can stop synthesizing the amino acids, and thereby you can kill the plant. So either to kill the microbes or to kill the plants without affecting the human being, yes, you can use it. That's what antibiotics are working, and some of the herbicides are working with inhibiting the plant growth. So you can choose it. You will see how the mechanism of Roundup. Have you heard of Roundup? The weedicide. You will see the mechanism. Okay. The amino acid biosynthesis. The amino acid biosynthesis, they need the solution for, the biosynthesis provides solution for very important biochemical mechanism. Solution for biochemical problem. That's very important. Okay, there are three key points. Which you should remember that. 3 key points. That's number 1. Fundamental problem for life on the earth is the fixing the nitrogen, and to use the nitrogen as a usable form. Usable form. That means amino acids. Okay, that's number 1. Who is doing this job? First job is done by bacteria. Okay. Bacteria can fix it. Okay. How? They put nitrogen, 3 bond, so N2, right? N2 is the symbol for nitrogen. 1 N, another nitrogen atom, another nitrogen atom. They bond with 3 bonds because their valency is 3, so they bonded, and they are so strong. Okay. Nitrogen, and this one will be cleaved to ammonia, and ammonia to amino acids to the plant, and even in the bacteria they can also synthesize from ammonia to amino acids to synthesize the bacterial protein. So the next one is the carbon backbone, where the carbon backbone is coming for the amino acids, that is coming from glycolytic pathway. Glycolytic pathway will provide the carbon backbone for the amino acids. And pentose pathway and TCA cycle, they also provide, or citric acid cycle, they also provide carbon backbone for the amino acid. So the ammonia is coming from bacteria, and the plant can also produce these amino acids from ammonia, and bacteria can also produce amino acids. But in the amino acid structure, you have a carbon, hydrogen, nitrogen. So the nitrogen is coming from the atmosphere, nitrogen, and through bacteria, and also the carbon for this is coming from, for a synthesis of amino acid, is coming from glycolytic pathway, pentose phosphate pathway, as well as TCA cycle. So that one should remember that. Okay. Now the next level is to understand the stereochemical, S-T-E-R-E-O, stereochemical. I want to refresh your chemistry, stereochemical control problem. Okay. How the stereochemistry is involved in controlling the synthesis of, that is, synthesis of isomer. Synthesis of isomer. What is an isomer? Do anyone know what is isomer? Isomer is, yeah, same molecular formula, right? And same molecular weight, same number of atoms and atoms of carbon, hydrogen and everything, nitrogen, but they have, structurally they are different. Structure different. How is it possible? I'll give an example. In isomer, the carbon is having 4 valency. Okay. Here the atom 1, another atom 2. Suppose this is a nitrogen or a hydrogen, or another one is acid group is there. The another one is a CH3 group. So another group, fourth another group. So 4 valency of this carbon is connected with 4 different groups or atoms. Then this particular carbon exhibit an asymmetric carbon. They are, it is not symmetrical arrangement, asymmetry. A-S-Y-M-E-T-R-I-C. Asymmetric carbon. This asymmetric carbon exhibit an isomer pattern. In other words, they can also, in a way, they can also express the optical rotation. If you keep like a left, they call it minus levo rotatory. R-O, this is one word, R-O-A, it's R-O-T-A, or rotary. And another one is the positive or the right side rotation, they call it dextro rotatory. Rotary or rotatory. So if you consider this part, the amino acid which is synthesized, they mostly, except glycine, glycine is a CH2, except glycine is not an asymmetry, but rest of the amino acids, they exhibit the asymmetric carbon, and that is responsible for the levorotary or dextrorotatory or the chirality. In other words, this characteristic we call it as chirality, C-H-I-R-A-L, chirality. Chirality, L-I-T-Y, C-H-I-R-A-L-I-T-Y, okay, chirality. And this one, which is exhibited by the amino acids, and that is from the bacteria can produce this type of amino acids, okay. And during this formation of these different amino acids, the very important enzyme is transaminase. Transaminase enzyme. And this transaminase enzyme is linked with different species and even proto-organisms. So they are evolutionary important. Evolution. They are linked with a prototype to the higher organism, the transaminase enzyme, and thereby the evolutionary climb that we are derived from bacteria. Okay, so that is a different story. Now, the third part of the key point one should remember in the biosynthesis, which I mentioned earlier, is the regulation. Okay? Regulation. How we regulate the amino acid biosynthesis. Okay? And the biosynthesis pathway may be switched on or off. How? Because of the demand of the inside the cell. Suppose the cell need a particular amino acid, then it will synthesize that one. Suppose if we synthesize, if it is keep on synthesizing, no. There is a particular limit. What will happen? The excess synthesis is avoided. Excess synthesis of amino acid is avoided. Okay? And it will inhibit the product of this amino acid synthesis will inhibit its own synthesis. In other words, suppose this is the amino acid that is produced, this is the reactants A, and AB give C, C to give D, and D will give you E. Okay? So these are the steps. These are the step 1, 2, 3, 4, 5. This is a step which will produce a particular amino acids, which is synthesized now. But you don't want to synthesize more and more and more and more, more and more, more and more amino acids. No. So once the demand is been satisfied by the cell, and the E will go in inhibit here or here. So it will inhibit its own synthesis, and this is called feedback inhibition. Okay, feedback inhibition of, because it's synthesis, the product of this amino acid itself, now it will inhibit its own synthesis. So this has been exclusively studied in the microbes and in the other tissues as well. Okay. So the next part is the nitrogen fixation. Okay, you have to see on the nitrogen fixation. That's in the bacteria. Nitrogen fixing. Nitrogen fixation into the bacteria. The bond, as I mentioned earlier, nitrogen, they have a triple bond. Okay, this is one atom. This is another nitrogen atom. This is a strong bond. They can the energy level stored in this bond is 940 kilojoules per mole. So this is the amount of energy. Okay, this bond is highly energetic, and this bond is highly resistant. Again, resistant to chemical attack. Chemical attack. So they are so strong. You need more energy to break this particular bond in a physical mean. There's one thing you can do. By strong lightning and ultraviolet radiation, they can break this bond. They can break this bond. But that is in atmosphere, they will produce more of nitrogen fixing by lightning, and then as well as the ultraviolet rays, they can break the bond. Otherwise, it is resistant to chemical attack. You cannot do any chemical attack to split the nitrogen molecule into nitrogen atom. So, the bacteria, they have a trick. Bacteria and archaea. Okay, bacteria and archaea. A-R-C-H-E-A-E-A. Archaea. And this is the one Dr. Sobolov is doing much of his work in his, with his student Matthew Boyd, who is now in Victoria. And he has done published some work with Dr. Sobolov to fix certain type of nitrogen fixing organisms and environmental importance. Different kinds of archaea they have identified now. So, coming to our point, how bacteria and archaea and they are fixing it. Okay. In the higher organisms, they are not able to fix the nitrogen. Okay, they are unable to. That the plant and animals cannot fix the atmospheric nitrogen. Okay, Only we have to do with the bacteria. So in the bacteria, there's one of the bacteria is called in the rhizobium, R-H-I-Z-O-B-I-U-M, rhizobium. And this bacteria, they invade the roots. They invade the roots of legume plants. You know, the beans, variety of plants, the L-E-G-U-M, legume plant. And they form in the root nodules. The bacteria, the rhizobium bacteria, present in the root, okay, in the roots, and they form nodules, root nodules, and these root nodules, they fix nitrogen there, or as a ammonia there. So they will instantaneously, they provide more fertilizers to the plant. That's why if you want to plant rice field, before they plant the rice seed in the land, the paddy field, they used to put legume plants to start with, and then after 3 months to these legume plant nodules, they fix atmospheric nitrogen to the soil. Then after 3 months, they harvest the legume, but the soil will enrich with ammonia and urea, and then they used to plant the paddy field. So in that way, this is a natural fertilizer, not from chemical fertilizers. It's a natural fertilizers using this particular plant. In other words, we can also fix the rhizobium in the lab, growing more and more, mixed with the carbon activated charcoal, and then you can sell it as a active organic fertilizers or biofertilizers. So that is also been possible. People are doing business with that one, nitrogen fixing bacteria, growing more and more and used for the fertilizers. Now how much they fix, how much these bacteria can fix the oxygen, how much? It is around 10 to the power of 11 kilogram. Imagine 10 to the power of 11 kilogram per year. From where? From atmospheric nitrogen. Okay, so this is the amount they are fixing it. This bacteria can fix it using the. They call it, these bacteria in a group call it diazotropic bacteria. The bacteria that can fix atmospheric nitrogen, we call it as a diazotropic bacteria. One of the bacteria is that rhizobium. You know, like rhizobium, there are azotobacter, there are different kind of bacteria are available, to fix atmospheric nitrogen. Depending upon the plant, we can use as a biofertilizers. And they will fix around 60% of Earth's newly nitrogen, or from the Earth. Newly produced nitrogen is fixed by this bacteria. Also the lightning, which I mentioned earlier, lightning and UV irradiation, they can also fix, because in the atmosphere you have nitrogen as well as hydrogen. So when the lightning pass through the air column with the nitrogen present, the nitrogen molecule is being split into two nitrogen atoms because of the lightning energy, and thereby they can also combine with the hydrogen. They can also produce ammonia at the time. The UV irradiation also more powerful, that will also break the triple bond between nitrogen atoms. So they will fix around 15% of the atmospheric nitrogen as a ammonia, this lightning and UV. Now the rest of them are 25%. 25% of the nitrogen, atmospheric nitrogen, can be fixed by industry to produce as a urea or the fertilizers. Okay, and this is being done by the scientist Fritz Haber, 1910. Why I am writing this 1910? Till after 100 years, still we are using the same method. We use the same method from atmospheric nitrogen to ammonia or to urea. So they used to do the nitrogen with the mixing with hydrogen, mixing with hydrogen gas, hydrogen gas, and the catalyst is iron. Iron is the catalyst, and they also used 500 degrees centigrade. See, imagine 500 degrees centigrade temperature, okay, and also the pressure. How much? 300 atmospheric pressure. So if you have a device with the iron, 500 degrees centigrade heating, and then 300 atmospheric pressure, this is the pressure. Under these circumstances, you can fix the nitrogen and the hydrogen from the atmosphere, and then you can produce more of ammonia. And that's for the industrial use. Even now they are using it. Okay, that's what the ammonia synthesis from the atmosphere, which we have studied now. The next step in the biological process, this is the which I mentioned earlier, which is a physical process. Now in the biological process, in the biology, what we need to do, nitrogen fixation by enzyme, okay, and the enzymes, we call it the complex enzymes, not simple enzymes, they are complex enzymes. Nitrogen is fixed by biological mean by using the complex enzymes, and they have a multiple redox center, reduction oxidation centers. They call multiple, not one, multiple redox, reduction and oxidation. So redox centers. So the multiple redox centers are present in these enzymes, and they will convert the nitrogen from the atmosphere and getting into ammonia. And they have got the name nitrogenase, nitrogenase, nitrogenase enzyme complex, nitrogenase enzyme, nitrogenase complex. And this enzyme complex is fixing the nitrogen, atmospheric nitrogen, and this nitrogenase complex is made up of two proteins, two enzyme proteins. There are two proteins which is involved, two proteins, and one is reductase. And this reductase provides what? It provides electrons. Provides electrons for reduction, right? For electrons with high reducing power. Electrons with high reducing power. Increased or high reducing power. And then another enzyme is nitrogenase. Nitrogenase enzyme, which I mentioned earlier. And this nitrogenase is the number second, the two proteins. The second one is the nitrogenase enzyme. And they use the electron. See, one enzyme will provide the electron, which will supply the electron. The another enzyme which will use this electron. Okay. And the supply to use, there is a transfer. Okay. The transfer of electrons done by transfer of electrons. How? The electrons are transferred. You need the energy for the electrons. Transfer of electrons done by ATP. Okay. So, what you need now? You need a reductase. Okay, number one, a nitrogenase and ATP molecule. If you have this enzyme unit and this enzyme subunit with the ATP, you can easily convert to nitrogen into ammonia. Okay. So you need the energy for that one, because you have to transfer the electrons into the useful one for reduction. So this nitrogenase, in other words, this nitrogenase enzyme, this complex enzyme is sensitive. They are sensitive to oxygen. So oxygen should not be there in the root nodules. Okay, when the atmospheric nitrogen is fixed by the bacteria, that's the rhizobium, and which has this enzyme, nitrogenase enzyme and reductase enzyme and the ATP, and they can fix it. But at the same time, that particular enzyme is sensitive to oxygen, meaning the oxygen can inhibit this enzyme complex, inhibit the nitrogenase. So you should not have this oxygen. Suppose this is a plant and this is the root system which is going on. So, and here is the leaves, okay, and the bean plant which is coming up. So these root nodules, you have the more of the bacteria, and normally you won't get the oxygen this much. However, these plant, the root nodules, and they have one particular leg hemoglobin. Hemoglobin. You know about the oxygen is highly affinity towards the hemoglobin. So here the legume plants, so they got LEGHEMOGLOBIN. Leg hemoglobin will capture the oxygen. Oxygen is being captured. So where this leg hemoglobin there, they are present in the membranes or the root membranes of the plant. So they will capture the oxygen present in the surrounding the roots, and thereby it will provide a congenial atmosphere or the optimum atmosphere, remove the inhibition of the oxygen, and thereby enhancing the nitrogenase oxygenase or nitrogenase activity, nitrogenase activity to fix the atmospheric nitrogen. So this is very important, leg hemoglobin which is present. And this is something like, just like a hemoglobin which is present in homology of our hemoglobin as a human, or any mammalian hemoglobin, which will capture the oxygen. So the iron molybdenum, okay, the nitrogenase enzyme, they have a cofactor, okay, cofactor, meaning they are the active sites of the enzyme, the cofactor. The cofactor for nitrogenase is iron molybdenum. This is another metal, MOL molybdenum, BDENUM, iron molybdenum complex in these one. And they actually use or reduces the nitrogen atom. They are the cofactor, which you remember. And the electron transfer, which is on the reductase as well as this nitrogenase enzyme, you have iron sulfur unit, that's called Fe, yes, Fe for iron, S for sulfur. They are a cluster, iron sulfur proteins. So they are involved in the electron transfer, and this will be useful in understanding the mechanism of how the nitrogenase and reductase enzyme transfer the electron. They are transferring through this subunit, iron sulfur subunit, which will easily be the carrier of the electron. In other words, the one enzyme to the another enzyme, the one part of the enzyme to the other, the iron sulfur unit are the proteins which will help transferring the electrons or for the reduction. By this process, we assume that we got ammonia is being produced. Ammonium ion with another form, NH4, ammonium ion. This is ammonia and this is the NH4 plus, which is called ammonium ion. Okay, you should remember that. So this is called ammonium ion, ION, ammonium ion. Okay. And they are associated with this ammonium ion, associated with the glutamate and then via glutamine. They react this ammonia with alpha ketoglutarate, alpha ketoglutric acid. This alpha ketoglutarate, which will combine with the ammonia, okay, NH4 plus, it will produce what? Glutamic acid, glutamate. Glutamic acid is the first amino acid can be produced. So the ammonia and ammonium ion, how they are produced from bacteria, and the bacteria will provide this ammonia, and then the plant or the other sources are inside the bacteria. The alpha ketoglutarate react with the ammonium ion to produce the glutamate, and then the glutamate will produce the next different type of amino acid combination with the other groups of keto groups. So the reaction of the ammonium ion plus alpha ketogluteric acid plus NADPH reaction plus H plus. Okay, these are plus. All of them are combined together, and they form a reversible reaction. I am writing that glutamic or glutamic acid plus NADP plus H2O. So this is the equation how the glutamate is synthesized in the ammonium. Okay. So this reaction involves like a Schiff base. This mechanism, just one example I'll give you. R1 and R2, this is a different group, and this is the carbon. This carbon valency 1, 2, and then 2 valency for nitrogen, and this is for the another valency for nitrogen is getting on. So nitrogen is 3 valency, carbon is 4 valency. Now this C double bond N, and this is called Schiff base. SCHIF, Schiff's base. Okay. So the formation of the Schiff base is the fundamental principle in synthesizing of any amino acids. So whatever the mechanism which you read in the book, they will go around with this. How a carbon and nitrogen is combined together. That is very important. As I mentioned earlier, nitrogen to ammonia it can synthesize. And then what? The ammonium or ammonium ion is joined together a carbon atom. And that carbon linkage is through the Schiff base formation. So that involves different steps, which is I'm not going to that detail. But please remember, after the ammonium ion, it linked with the carbon through the formation of the Schiff base. That is enough for you to understand, because the carbon and nitrogen bond is very critical, how they are linked to each other. Now, the next one is the amino acids are made from intermediates of TCA cycle. Amino acids, they are made from intermediate, as I mentioned before, intermediates of what? TCA cycle, tri-carboxylic acid cycle or citric acid cycle, and other pathways. So all of them are intermediate. So that's why this class or the course is the intermediary metabolism. So we have to know about what is the intermediates or the amino acid, or what are the intermediates of TCA cycle that will produce of these amino acids. For example, if you know about the oxaloacetate, OAA, I'm just writing an abbreviated form of oxaloacetate, which will produce aspartic acid, aspartate, ASP, aspartate, and this aspartate will give rise to aspargine, okay, ASN, aspargine, and then the aspartate will give you methionine, MET, methionine, and the aspartate will give you, will produce lysine, LYSIN, lysine, and the aspartate can also produce threonine, THR, threonine, and the threonine will produce isoleucine. So if you remember, the oxaloacetate is one of the intermediate for the TCA cycle, but if you get excess of the oxaloacetic acid, and they will, by transamination reaction, getting aspartate will have a, the basic for the synthesis of aspargine with another amino group together. So, and it can also produce a sulfur containing methionine, and it can also convert it to lysine. This aspartic acid convert to threonine, and the threonine is the base for the synthesis isoleucine. So this is one pathway. The another pathway, the phosphoenol pyruvate, which you know about the glycolytic pathway, and erythrose 4-phosphate. Erythrose, ERYTHROSE, erythrose 4-phosphate. The combination of these two will produce another compound like another amino acid, phenylalanine. Phenylalanine. And the phenylalanine will produce tyrosine molecule. And now it can also produce directly tyrosine molecule, and it can also produce tryptophan, another amino acid. If you see, these amino acids are aromatic amino acids because they are the like aromatic carbon, like this, benzene ring, and also you have this tryptophan have this structure, and tyrosine will have the structure of the hydroxy group, and then another one is for the without hydroxy group with the phenylalanine. So all these are the aromatic amino acids. Aromatic amino acids are derived from phosphoenol pyruvate and erythrose 4-phosphate combination. You will get this. So the amino acid biosynthesis, you should remember how they are produced, which amino acid. Aliphatic means a long chain, straight chain, there is no circle, or the ring structure. That aliphatic amino acids, that is different. That's what I mentioned earlier. One of the TCA cycle, here the oxaloacetate, aspartic acid, all of them are the linear molecule or aliphatic amino acids. And the aromatic amino acids are derived from phosphoenol pyruvate erythrose 4-phosphate. Now, the other one is the aliphatic again, pyruvate. Pyruvate is an intermediate, and it will produce more of alanine, and can also produce valine. It can also produce leucine. So pyruvate is the intermediate on the carbohydrate metabolism or the glycolysis metabolism. So where you can produce of alanine, valine, and leucine. Okay, this is the one part. And then the alpha ketogluteric acid, which I mentioned earlier, which will by transamination it will produce of glutamine. Okay, glutamine, another amino acid, glutamic acid. Glutamic acid will produce GLN, that is glutamine, and then it can also produce proline, and it can also produce arginine. So arginine is synthesized from glutamine, and proline is synthesized from glutamic acid. This glutamine is coming from the glutamic acid. The alpha ketoglutarate, by transamination reaction, this is a glutamate or glutamate. And this glutamic acid or glutamate is one amino acid that will produce arginine, another amino acid that will produce proline, another amino acid that can also produce glutamine. That's another GLN. That's also another amino acid can produce. Now the next amino acid that is on histidine, that is derived from ribose. Ribose 5-phosphate. This is the pentose phosphate pathway, and it will produce histidine. This is another amino acid, histidine. Ribose 5-phosphate will produce histidine, and the another one that's on the 3-phosphoglycerate. This is also we studied in the glycolysis, glycerate. We call it the PGA, glyceric acid, phosphoglyceric acid, PGA. Okay, and it will produce serine or serine. And the serine will produce cysteine, cysteine. Cysteine, and another one is the glycine. It can produce glycine. So glycine and cysteine produce from serine, and serine is produced from 3-phosphoglycerate, a 3-phosphoglyceric acid, and histidine is produced from ribose 5-phosphate. Okay. So the next one is we have to remember the basic amino acids. Already we see in the amino acid or the protein metabolism. Basic amino acids which we have studied, there are 20 in numbers. Okay, there are two groups. They have non-essential and essential amino acids. Non-essential amino acids, they are listed as because they can synthesize in our body. So it is not necessary that we have to get it from the diet, because our body can synthesize. What are these non-essential amino acids? Alanine, arginine, aspargine, and aspartic acid, ASP, and then cysteine, CYSTEINE, EINE, cysteine, and glutamate, glutamic acid, glutamate, and glutamine, GLN, glutamine, and then I write it here, glycine, and then proline, and serine or serine, and tyrosine. All of them are non-essential amino acids. Now, I will write up the essential amino acids that is derived from diet sources: histidine, isoleucine, and leucine, and lysine, and methionine, and phenylalanine, and threonine, threonine, and tryptophan, and valine. I write it here, valine. So these are the amino acids we have to take it from our diet. If you are not taking these essential amino acids in your diet, then you will suffer from protein malnutrition. PMN, protein malnutrition. So you are suffering from protein deficiency, and because you cannot support your body with these amino acids, because for our body function we need all these amino acids. So dietary protein is very important. So the next one is we have to go for what is the importance of methionine and the individual, the essential amino acid. In other words, the mechanism of SAM, yes, adenosyl methionine in a group. Okay. So what I'll do, I will give you a break now. Come back another 10 minutes, and then we will continue on the rest of the class now. Okay. So this one we have to do about the yes, adenosyl methionine. Okay. SAM is an abbreviation for yes, adenosine. That the sulfur atom is attached to adenosine group, ADENOSYL, adenosyl methionine, methionine. Okay, so yes, AM we call it as a SAM, yes, adenosyl methionine. This is a very major methylene group donor. S-SAM or S-adenosyl methionine, probably you might have studied in your earlier biochemistry class for the addition of methyl group, CH2, or methylene group. To add, we have to use the S-adenosyl methionine. Is very important. They are the carrier, or they will transfer the methylene group from one amino acid to the other, or from the one compound to the another molecule. So it forms a donor, methylene donor. Okay, how that will help to convert methionine into homocysteine. Okay, so we are going to see how the methionine is converted to homocysteine using this SAM. So that's the next step. Okay. Met means methionine plus ATP molecule, okay, and they will produce SAM. So SAM is synthesized with ATP, the ATP and the combination of methionine to form, because the methionine is being adenosylated. In other words, this is being energized methionine. If you think that methionine is an essential amino acid, it is an essential amino acid that is coming from diet. If you don't have the methionine in the diet, and you will suffer from the deficiency of some of the amino acid like homocysteine. Okay. Now coming to the story, methionine activated with ATP to form SAM, and now the S-adenosyl methionine will get like a RH, the one other group, which will remove the methylene group, or CH3 and H+. See, RH, this is the alkyl group, which will remove the methylene group from here, methionine, and thereby it's removed from this, and it forms S-adenosyl homocysteine. S-adenosyl homocysteine. S-adenosyl homocysteine, and this is again on hydrolysis with water molecule removal of water. It will it will add water the other way around. Add water to it, hydrolysis. Add water to it, and that will remove adenosine. Okay, if you add water to adenosyl homocysteine, that will remove adenosine, and then you will get homocysteine. Homocysteine now is produced, and this homocysteine, it will, in the presence of the tetrahydrofolate, in presence of N5 methyl tetrahydrofolate. If you do this, and you add this tetrahydrofolate, or with react with the tetrahydrofolate, N5 tetrahydrofolate, it will release tetrahydrofolate because the methyl group is being added to it, and thereby methionine is being produced. So this is a cyclic process, how the methionine is involved in synthesis of the homocysteine, and in other words, the homocysteine can also be, with the addition of methyl group getting into the methionine again. So one way it is loses and another way it gains. So this is a cyclic process with the help of tetrahydrofolate. In other words, we can also write in a different way of the cycle, like a SAM can produce, and the SAM will produce what? S-adenosyl homocysteine, where the active methyl group is being removed. Active methyl group, CH3 group, is removed from this, and then S-adenosyl methionine on hydrolysis will produce homocysteine, and the homocysteine that's on with water. Adenosine is being removed from this, and then by methyl addition, methionine, by you are adding the active methyl group or methyl group by N-methyl group, methionine, and from methionine, if you add ATP molecule, it will produce the SAM. So SAM plays a major role. S-adenosyl methionine. In other words, it will provide the active methyl group to other amino acids or other carbon group. It will add the SAM. So that's very important. The role of amino acid, how they donate methyl group using the SAM. That's very important. So the next one, the cysteine is synthesized from serine or serine. But cysteine, C-Y-S-T-E-I-N-E, cysteine, it is synthesized from serine, C-E-R-I-N-E, serine, and homocysteine. See, the previous one I showed you from, we discussed about the methionine to homocysteine. Now the homocysteine and serine to produce cysteine. Right, after homocysteine HC plus serine, and it will produce cystathionine, C-Y-S-T-A, cystathionine, T-H-I-O-N-I-N, cystathionine, and the cystathionine, during this process, it will eliminate water. Homocysteine serine combination, which will release water and to form cystathionine, and the cystathionine on hydrolysis, if you add hydrolysis to on water addition, cystathionine, it will release the ammonia group or ammonium ion, and it will form alpha ketoglutaric acid. And alpha ketoglutaric acid plus cysteine, cysteine, plus cysteine. Okay. So the cysteine formation from serine and homocysteine. Okay. So this is the pathway, and this the very important pathway is the intermediate is cystathionine. So cystathionine synthase enzyme or mutation on this, and you will not get the cysteine. So in other words, if the homocysteine is not being used up by serine, what will happen? You get high level of homocysteine. Okay, that is in the high levels of homocysteine. If you have the high level of homocysteine, that lead to high risk of coronary heart disease. Coronary heart disease. CHD, coronary heart disease. Why? The homocysteine is not being used with the serine to form cysteine. So you get this coronary heart disease. And this is a high risk factor for coronary heart disease, meaning before the disease is onset, if you measure the blood level of homocysteine, if it is increased abnormally when compared to the normal level, then you can say that patient is going to get coronary heart disease soon. So that is the diagnostic marker. Okay. Now, this how it will promote, because the homocysteine that will promote the vascular smooth muscle cell proliferation. Okay, and thereby it will be proliferation. So they are present in the heart. Okay, these vascular smooth muscle cells will produce more and more, and that will block the blood flow, and thereby you get the CHD or the coronary heart disease. Then the patient should undergo a bypass surgery to remove this artery and then put another artery there. So that is a big problem again. Okay, blood wall thickening. In other words, the blood wall thickening because of the homocysteine accumulation in the blood. Also, it increases the homocysteine. Also increases the oxidative stress. Oxidative stress. It increases the oxidative stress, the homocysteine. And what is the treatment for this one? Homocysteine. The treatment mainly with the treatment with the vitamins. Okay, to increase the enzyme, boosting the enzyme synthesis, enzyme reactions. Okay, treatment with vitamin. So vitamin plays a major role to prevent the heart disease. In this case, pyridoxal phosphate. Pyridoxal phosphate, and they are the vitamin B6. Okay, and it is necessary for for cystathionine beta synthase. Cystathionine beta synthase enzyme. So this enzyme, which I mentioned earlier, homocysteine and serine combination, that needs the cystathionine beta synthase. So even these enzymes are there, but if there is a deficiency of vitamin B6, then that particular person lead to coronary heart disease because the accumulation of homocysteine in its blood. Okay? Even if the cystathionine beta synthase mutation, this enzyme will not be active, though you have a plenty of vitamin B6, then also they will get the coronary heart disease. Okay. In other words, vitamin B12, cobalamin, vitamin B12 also support the methylation of homocysteine to methionine. So as I mentioned earlier, homocysteine to methionine. So this pathway, it also involves the methylation. This methylation is done by the vitamin B12. So it will be easier. So more of homocysteine is converted to methionine. If you don't have the vitamin B12 in the blood, then again the accumulation of homocysteine is being prevalent. So you have to have the balance of the homocysteine in the blood. Okay, now I want to go in the aromatic amino acids. that is true, that is for phenylalanine and tyrosine and tryptophan. And for this synthesis, there is a very important steps which is involved in the synthesis through shikimate, S-H-I-K-I-M-A-T-E, shikimate and chorismate, C-H-O-R-I-S-M-A-T-E, shikimate and chorismate. So you have to remember that they are the intermediate which will help to synthesize the phenylalanine, tyrosine and tryptophan. And they are predominantly present in the in the bacterial system, and also the biosynthesis of some other plants. So they are the intermediates. So in the intermediate metabolism on this class, intermediary metabolism, these are the very important intermediates which synthesize the very important amino acids, shikimate and chorismate. And they are the essential essential amino acids synthesizing in bacteria. Okay? So, we'll see how that will happen. As I mentioned earlier, phenylalanine, tyrosine, tryptophan, okay? They are synthesized through a common pathway. There's a common pathway that will involve from first step is condensation. Condensation of phosphoenol pyruvate acid plus erythrose 4-phosphate, which I mentioned earlier in the lecture. Phosphoenol pyruvic acid and erythrose 4-phosphate, and they will produce together a 7-carbon unit, okay, 7-carbon but open chain unit, not a closed ring, open chain unit, and they form into, after oxidation, the oxidation step. Next step is oxidation, and also removal of phosphate group, and that will cyclizes the 7-carbon unit to 3-dehydro, I write it here, that is very dark, dehydro, Q-U-I-N-A-T-E, quinate, dehydroquinate, and they dehydroquinate on the removal of water is removed, or dehydration step, it will produce 3-hydroshikimate, S-H-I-K-I-M-A-T-E, 3-hydroshikimate, and the 3-hydroshikimate on addition of NADH and H plus, it will change to shikimate, and the shikimate on phosphorylation, it will produce shikimate 3-phosphate on phosphorylation, and this one again on the shikimate phosphorylate with the phosphoenol pyruvate again, okay, and it will produce the 5-enol pyruvate intermediate, 5-enol pyruvate intermediate, and this will produce chorismate, C-H-O-R-I-S-M-A-T-E, and this chorismate, the common precursor for this compound, phenylalanine, tyrosine and tryptophan. So all of them is coming from the shikimate and chorismate, but the stepwise process, phosphoenol pyruvate with erythrophosphate, the starting point, it will produce a 7-carbon, because this is a 3-carbon unit, this is a 4-carbon unit. They are combined, joined together to form 7-carbon open chain, On oxidation, they will cyclize to 3-dehydroquinate, and that is on removal of water or dehydration, 3-hydroshikimate, and then a reduction process with NADH and H+, shikimate, and then shikimate phosphorylation to produce shikimate 3-phosphate, and then phosphoenol pyruvate to add to it, and then 5-enol pyruvate intermediate, and then that form chorismate, 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 and this is the intermediate, a common intermediate for phenylalanine, tyrosine, and tryptophan. Now, what happened from chorismate now? This is the next step which is involved in this one. Okay. Before I go into that, I want to, I will just get into the glyphosate. Okay. That is the importance of this pathway. The chorismate pathway is the glyphosate, not glyphosate. It's a glyphosate, and this is something like a Roundup. Roundup you normally do in a weedicide killer, a broad spectrum of a herbicide, and this has become a nuisance now, and there are a lot of environmental pollution which is going on, not to use the Roundup or use the Roundup or something, because some of the plants, they are resistant to herbicide or glyphosate, and they are the uncompetitive inhibitor, inhibitor for what? The 5-enopyruvate shikimate 3-phosphate enzyme. The enzyme, this is the inhibitor for the enzyme which involves the 5-enol pyruvate, chorismate. This particular enzyme is being inhibited by the Roundup or the glyphosate. Okay, and it blocks this particular inhibitor, right? The glyphosate. It inhibits what? It inhibits the aromatic amino acid formation. Aromatic amino acid formation. What are the aromatic amino acid? They are the phenylalanine, tyrosine, tryptophan. So all of them, they are being blocked because of the inhibitor of the Roundup or the glyphosate, and thereby it can inhibit the herbicides or the weeds or there are some plants, but at the same time it is not harmful to animals. That's why we can use it. Animals, fish and dogs and other things, and it won't interfere or this won't inhibit because the enzyme is not there. Now we will go into the mechanism. I mentioned earlier chorismate, CHORISMATE. Chorismate is the intermediate, and how this chorismate is being involved in the synthesis. One side, it will produce of a prephenate, 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 and this prephenate will produce, in one side, phenyl pyruvate, and this phenyl pyruvate will produce phenylalanine. Phenylalanine, this is the amino acid, and then the prephenate, which will go para-hydroxyphenyl pyruvate. So the para-hydroxyphenyl pyruvate now will produce tyrosine. Now the chorismate, another pathway, and this pathway will produce anthranilate. Anilate. I put anilate, ANILATE, anthranilate, one word, and this on PRPP, phosphoryl pyrophosphate, and removal of phosphate. This being added and removal of pyrophosphate, PPI, which will produce N-phosphoribosyl anthranilene. I'll write it in another paper, that's easier. Chorismate. Okay, chorismate from here anthranilate, anthranilate, and then the anthranilate on the PRP, phosphoribosyl pyrophosphate, and removal of pyrophosphate, and it will produce the N, or nitrogenous phospho. In nitrogen group, the phospho group is added. Phosphoribosyl, R-I-B, ribosyl, phosphoribosyl anthranilene, anthranilene, which is the intermediate again, and then it will produce the 1-deoxyribose 5-phosphate, 1-deoxyribose 5-phosphate, and on decarboxylation, the carbon dioxide is removed from this step, decarboxylation, it will produce indole, indole 3-glycerol phosphate, one compound, indole 3-glycerol phosphate, and then it will produce indole, indole, and then if you remove water and you add serine, serine amino acid is being added to it, so you got the indole. Now the glycerol phosphate, carbon dioxide is removed. Yes, indole 3-phosphate, and then it will produce to indole, and then I'm sorry, here it is not coming up here. Okay, indole 3-phosphate, and then this indole 3-phosphate to glyceraldehyde, right? The glyceraldehyde phosphate, it is being removed here. Glyceraldehyde 3-phosphate, carbon dioxide removal, glyceraldehyde 3-phosphate is being removed from here, and that will glyceraldehyde 3-phosphate to indole is being produced here. I'll write it here. I'm sorry, that one here. I write it from the step indole 3-phosphate, and this indole 3-phosphate removal of glyceraldehyde 3-phosphate, and then that will produce indole. And then from this indole, you have to, if you add the indole, add serine amino acid and removal of water, and that will produce tryptophan amino acid, tryptophan. So it is a different steps, which is from indole 3-phosphate to produce tryptophan. In other words, as I mentioned earlier, this charismate anthranilate to indole 3-phosphate, and the charismate will produce the phenylalanine, tyrosine, and these steps, intermediate step, various steps to indole, and that will produce the tryptophan. So the charismate is the common intermediate for producing the phenylalanine, tyrosine, and tryptophan, or any aromatic amino acid. So please remember, the aromatic amino acid biosynthesis involve charismate, and then before the charismate there's shikimate, and all of them are both of them are intermediate in producing this amino acid, these 3 amino acids. Now, not only this, the amino acids are precursors for other biochemical molecules or biomolecules. Okay, that's the next step. What is the use of this amino acid? We have studied about the essential amino acid and non-essential amino acids and all of them. So the use of this amino acid, it will produce of the other biomolecules, biosynthesis. What are the other biomolecules? Adenine, and then cytosine. Say adenine, cytosine, they are the building blocks for DNA synthesis, okay? And sphingosine, sphingosine, sphingosine as a phospholipid, and the histamine, one of the mediators of inflammation, histamine, And then thyroxine is a hormone. Thyroxine, a hormone, right? Tetrahydrothioronine. And also epinephrine, another hormone. Epinephrine. Epinephrine, I-N-E, epinephrine. And serotonin, S-E-R-O-T-I-N, serotonin, okay. And the nicotinamide, NADPH and NAD, you have studied that, nicotinamide, nicotinamide. All of them are the biomolecules. For the synthesis of the biomolecules, you need more of amino acids. So you should be very careful in the amino acid metabolism. Why we need to study this amino acid synthesis? Because amino acids is the building blocks of other biomolecules. The next step is in the understanding of the biomedical importance is the porphyrins. Porphyrins. It's very important. The porphyrins are synthesized, they are synthesized from, this is another biomolecule, and it is very important in the heme metabolism or the heme biosynthesis, which is involved in the oxygen transport in our body. So porphyrin is the building blocks for the heme, and then how the porphyrin is synthesized from amino acid. Porphyrin is synthesized from glycine and succinyl-CoA, glycine and succinyl coenzyme A. So succinyl acid or succinyl coenzyme from A from the fatty acid, as well as from the amino acid, glucose or TCA cycle. It can also produce from the the glycine and succinyl-CoA. They combine together to form porphyrin. How? They combine with the presence or removal of carbon dioxide and removal of coenzyme A. This is also being removed from this when the combination. It will produce of a delta amino levulinate, L-E-V-U, levulinate, okay, delta amino levulinate. And this delta amino levulinate, not one molecule, there are 8 molecules. Here, other papers. The 8 molecules, okay, 8 molecules of delta amino levulinate. Okay, I put delta amino levulinate, and they actually remove 8 hydrogen atom from this, and 16 water molecules, H2O, is being removed to form propionate. And there are the propionate, again they form as a 4, okay, porphobilinogen, P-O-R-P-H-O, porpho, B-I-L-I-N-O-G-E-N, 4 porphobilinogen, okay, and they are the linear molecule or the tetra. They are the linear molecule. That's a straight line. There's no circle here. But tetrapyrrole molecule, P-Y-R-R-O-L-E, pyrrole molecule, and they will produce a uroporphyrinogen. I'll write it here. Uro porphyrinogen 3, uroporphyrinogen 3, and they will produce coproporphyrinogen 3, coproporphyrinogen 3, and this will produce protoporphyrin, and this protoporphyrin with iron molecule or iron atom will form heme. So now the heme is being synthesized from the glycine and succinyl coenzyme A. Now, the heme is the one which is responsible for the oxygen transport or hemoglobin and different pathway. So now, the heme is being converted to biliverdin, the heme, or the heme catabolism. This is a heme biosynthesis, but the heme plays a major role in the medical practice, like heme catabolism, or it will remove two oxygen. Oxygen is added to it, oxygen with oxygen, and it will remove carbon dioxide, two carbon dioxide and plus water and NADH. Also, it will remove the iron from this. The heme is metabolized, it's been broken down, and then it will produce biliverdin. It will produce biliverdin, 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 and this biliverdin with the presence of NADPH, and which H plus, which will produce NADP plus, and this will produce bilirubin. So what is the importance of the heme? Heme is the precursor for biliverdin and bilirubin. Okay, they are the pigments. They are the colored pigments, and this heme degradation product. Heme degradation products of bilirubin and biliverdin, and they form a dark brown color. And this is the color whenever you get an injury, a bruises color. If you have a bruise or injury or anything, scratch and everything, at the time you are exposing the heme on the blood or hemoglobin exposed to the oxygen, and that will lead to degradation of the heme in presence of the oxygen. That will form a dark brown color in the bruises. Okay, that's the color of the bruises. Whenever you have the hurt or anything, you get that change in color, brown color, dark brown color, from red color to dark brown color, because of the formation of the products, degradation product of bilirubin and biliverdin. Okay. Now, the condition, if the porphyrin accumulate, if the porphyrin is not converted to heme in case of heme is not synthesized. So what will happen? The accumulation of porphyrin, that will lead to some inherited disorder, some enzyme deficiency, inherited disorder, inborn errors of metabolism. That is called the porphyria, P-O-R-P-H-Y, porphyria, R-I-A, porphyrias. And these are the deficiency of the enzyme in heme biosynthesis pathway. So that's why the porphyrias is occurring. So the porphyrin synthesized in, generally the porphyrin is synthesized in erythroblast, the red blood cell, erythroblast. Erythroblast synthesize the porphyrin, okay, and also liver. Liver can also synthesize the porphyrin, both of them, porphyrin. So if any liver disorder or in erythrocytes, the synthesis, suppose if you have a low hemoglobin content in the blood, what you call it, hypohemoglobinemia, some of the loss of blood, and it will take a while to produce more of the hemoglobin. There are certain anemic conditions. Erythroblast will have a deficiency in synthesizing the porphyrin, and thereby the heme synthesis is being blocked. Or in the liver disorder, some of the jaundice or any other liver infection, the porphyrin synthesis totally being affected, and thereby you get some of the disorders on that. So you have to be very careful on treating the porphyria when you have a deficiency. In the congenital erythropoietic porphyria, this is an inborn error of metabolism. Congenital means inborn. Congenital erythropoietic, the RBC production, porphyria. So this is a genetic disorder. The premature destroyed the RBC. The RBC is being destroyed prematurely. It is not being synthesized fully. And the accumulation at this time you have an accumulation of uroporphyrinogen. Accumulation of okay, porphyrinogen 1. Accumulation of uroporphyrinogen 1 and coprophyrinogen 1, that will lead to excrete in urine. Excrete in urine. So if it excreted in urine, also urine become red color. So red color urine. So that would be dangerous. People think that, oh, urine going with the blood, but that is a bit different because of the uroporphyrinogen color. And then in the teeth of this patient, if you have a fluorescent in the UV, fluorescence, if you treat with the UV irradiation to the teeth of the patient, the teeth, because of the accumulation of the porphyrin, and they also exhibit a strong red color. So teeth become a strong red in color because of the accumulation of this pigment of porphyrin. And then the skin of the patient is so sensitive to light. So light sensitive, this patient, okay, and the photoexcitation of the porphyrin, because the porphyrin accumulate in the skin, and that will photoexcited, and that will give a skin sensitive or light sensitiveness for the skin. And the the delta aminolevulinate and the porphyrinogen, and they, the accumulation of porphyrinogen and delta aminolevulinic acid. So both of them, they induces abdominal pain, because in the accumulation in liver, abdominal pain, accumulation of porphyrinogen and aminolevulinic acid in liver, that will lead to abdominal pain, and also neurological disorder. So in other words, they become mad. That's why the madness of George III of England. If you remember, if you study in your history, madness of George III England, and during the American Revolution, and this guy is become mad. And that madness of George of England is due to the accumulation of porphyrinogen or the aminolevulinic acid in the liver. So you have to remember certain disorder, amino acid disorder in this class. So with this, I'm finishing it up for your biosynthesis of amino acids, and then the next week we will see about the nucleic acids of DNA and RNA. I think that's according to your syllabus, and then we can go into for some drug development. Okay.